Hi! I've always wanted to be an astronaut, so I've always been pretty concerned about how astronauts stay healthy. After all, nobody wants a cold all the way to Mars and back. But being in space makes astronauts' immune systems weaker, so it's more likely that they'll end up getting sick. In 2015, I came across a website that asked high school students to design DNA-based experiments to run on the International Space Station. And the first thing I thought of was questions about the immune system. Why do astronauts' immune systems get weaker in space? And then what can we do to fix it? After doing more research, I learned that the immune system has cells called T helper cells that guide its response to pathogens. These T helper cells start out as naive or undifferentiated cells, and then based on signals from the rest of our body, they turn into either Th1, T helper 1, or Th2, T helper 2 cells. Each of these cell types is responsible for a specific kind of immune response that it triggers by producing unique signaling molecules called cytokines. For this immune response to work properly, there has to be a balance between Th1 cells expressing Th1 cytokines and Th2 cells expressing Th2 cytokines. But studies done on space shuttle flights with mice in the early 2000s demonstrated that even really short exposure to microgravity was enough to cause an imbalance in the ratio of Th1 to Th2 cytokine expression. Scientists then thought that the same thing might be happening in humans because they found that astronauts who had just returned from space had a lot more Th2 cytokines in their blood when compared to the blood of people who had stayed back on Earth. Based on this information, I hypothesized that going to space was messing up how the Th1 and Th2 cells were expressing their cytokines. If astronauts' Th1 cells and Th2 cells weren't expressing the right amounts of the right cytokines at the right time, that might explain why our immune response wasn't working properly when we're not on the planet. So for my Genes to Space experiment, I decided to use a special kind of PCR to look at the expression of certain cytokine genes to understand if the T helper cells were expressing the same cytokines in space as they were on Earth. PCR was the ideal way to answer my question about cytokine expression in T helper cells because it allowed me to zoom in on just the genes that code for specific cytokines. You see, I wasn't interested in knowing if the genes were there. After all, almost every cell in our body has the same DNA. Our skin cells, our immune cells, our muscle cells, it's all identical DNA. The reason that different cells function differently is because as cells differentiate, the genes that they don't need are turned off. So even though Th1 and Th2 cells have all the same DNA, so they have all the cytokine genes somewhere in there, Th1 cells will only express Th1 cytokines, and Th2 cells will only express Th2 cytokines. So I didn't want to use PCR to look at the Th cells' genomes. I wanted to look at this difference in expression, and to do that, I looked at something called the epigenome. The epigenome is a layer of chemical markers that goes on top of the DNA, like methylation, that controls how it's expressed. See, if your genome were a book, the DNA would be the sentences that you're reading, and the methylation would be like lines of pencil crossing out the sentences that you're not meant to read. Luckily, there's a kind of PCR called bisulfite PCR that lets you see if the DNA sentence that you're looking at is crossed out or not. Now, there's one more important fact that brings my whole theory together, which is, in different, totally unrelated cell types, scientists had found that going to space altered methylation profiles. So I'm thinking, what if it's the methylation going wrong and causing these T helper cells to express the wrong cytokines in space? So I proposed to look at that. I wanted to take Th1 and Th2 cell samples, keep half on Earth and send half to space, and then once the cells in space had been exposed to microgravity for long enough, I wanted to use bisulfite PCR to see if the genes for Th1 and Th2 specific cytokines were methylated differently, suggesting that they were being expressed differently in microgravity compared to the cells we had kept on Earth. If I saw that the methylation patterns of cytokines in space were different than on Earth, for example, maybe in microgravity, the Th2 cells were acting normally, but the Th1 cells weren't expressing enough of their Th1 specific cytokines, but were suddenly expressing some Th2 specific cytokines, That would help confirm my hypothesis that aberrant methylation profiles were leading to weird levels of cytokine expression in space that might even explain why we're seeing immune system dysfunction in astronauts. Maybe, if we could find a common pattern of space-induced changes in methylation, we could even use similar PCR tests to monitor astronauts' genomes before they started experiencing symptoms of space-induced immunodeficiency. But most of all, I wanted to do this experiment because it seemed like an amazing opportunity to use a cool molecular biology tool to learn more about how our genes work in space.